goes, but as Dr. Mack, we're going a little out of order, so Dr. Mack, I'm sort of putting you on the spot, and I appreciate it, but I just want to say how much I appreciate Dr. Mack. He is currently the director of the Department of Ophthalmology at the University of Montreal and left Edmonton. I'm not sure if that's because of the recent Stanley Cup or not, but he does hold the named chair there, and he first trained in medical genetics and then in ophthalmology in Canada. And he's been working with patients and families affected with choroideremia really since the beginning in the 1980s. So he's a young father, I'll just say it that way. And I'm grateful because when I first started with the Choroideremia Research Foundation, he was an active member of our scientific advisory board, a very robust and welcomed member, and now he's an emeritus member of that group. And so thank you, and Dr. Mack.
Thanks very much. Thank you all very much. And I really enjoyed Dr. Porter's introduction to genetics. It was amazing. I don't think I would be as fearless as he was in trying to present that material. So I can see my slides over there, so that's good. Excellent. So the summary of my talk is that there's an intronic variant that can be treated with an antisense oligonucleotide. So what does that mean? So the coding sequences of the gene are the exons. The intervening sequence in between the exons is thought to be not important, but it's very important as the gene becomes read to make the messenger RNA. And so in this intervening sequence, it's called an intron. In some families, in 3 families in Canada, and some of the family members are in the United States, there is an intronic variant that we can treat with an antisense molecule. So and I'll walk you through that. So as just to review, choroideremia has different variants, and they all result in a relative absence of the Rab escort protein 1. It's interesting that Dr. Porter is talking about getting a level of expression up to 20 or 50%, saying that might be therapeutic. I think it's much less than that. I'll go after that later. So the subretinal and intravitreal therapies have had their problems with inflammation. If we were to use something different than an something different than an AAV vector, to use an RNA therapy, it might be safer. Now, many of us were vaccinated with a product from Moderna and different pharmaceuticals that would hopefully make an immune reaction that would prevent you from being infected by the COVID virus. So these therapies are safe, and there are some anti-vaxxers. I'm sure there are a few in the audience, but I would propose that vaccination is an important thing to undertake. Okay, so viral vectors can trigger inflammation, and here's a little bit of a cartoon. I like the analogy with a cartoon. So the retina and the subretinal space are thought to be immune privileged. It's not an immune incompetent space, though. There are resident microglia. These are cells that are arrayed in the neural retina that are there to protect the retina from inflammation. And these cells, the microglia, become activated, especially when there's inflammation and especially if there's an immunogen, such as a viral particle. They begin to change shape, they become amoeboid, they move, and they also are designed to attack and phagocytize and create and secrete protein factors that will draw other cells into the retina. And so these cells are phagocytic, and they're there to prevent inflammation and to deal with inflammation and infection. But an antisense molecule, an RNA-like molecule, a highly modified RNA, is a very stable molecule. The adeno-associated viral vectors are stored at -80. They're shipped at -80 in a carboy for delivery through a pharmacy for use in surgery. But the RNAs are stable at room temperature and very stable for a long time. And they can be used with an intravitreal injection, so an injection into the eye, into the vitreous cavity, and without significant inflammation. Why am I saying this? Well, I didn't really disclose some of my conflicts of interest. I happen to serve on the Data Safety Monitoring Board of the ciprofloxacin trial, which is used for the treatment of Leber's congenital amaurosis, the CEP290 form of Leber's congenital amaurosis, a different form of Leber's congenital amaurosis than is treated with Luxturna. And there had been no significant inflammation with the use of this injecting into the eye. So Dr. Porter again did some very good description of the sense of exons in intervening sequences and the coding sequences of the gene, the use of the splicing factors that are there to create a messenger RNA. And essentially we are trying to clip the exons together to make a complete message. But the intervening sequence has code in it as well. And if there's a code that creates an additional splicing site, then it can include a pseudoexon, an exon that's not part of the proper reading frame of the gene. And if that happens, then it throws everything off, and the individual does not make a normal protein. So what would happen if we used a highly modified RNA, such as an oligonucleotide? It can bind to this site within the intron so that the splicing site sequence and the spliceosome doesn't recognize that target. And so think of it as Scotch tape right over the top. So the coding change is not sensed by the splicing machinery, and the exons are actually assembled properly. So this particular change in a gene, in the choroideremia gene, is in between exon 9 and 10, and we can use a highly modified RNA to prevent the splicing defect, and it will create a normal full-length Rep1 product. So here you can see on the top, this is a Western blot. This is looking at the protein that's being made. You have Rab escort protein 1. This is the normal gene product. It's a protein. The biochemistry is very interesting. And you can see here in lane 3, this is an individual with choroideremia and has a certain variant that you can see there. And you can see there's no red band. So there's no protein being made. In lane 4, you can see that red band appears because you've used an RNA, an antisense oligonucleotide, to treat the cells, and you make a normal protein. It's the same protein product as the control lane, which is lane 2. So what happens with the biochemistry? Well, in fact, we've been shown that the degree of prenylation, this is the lipid modification of the Rab proteins. So Rab escort protein 1 acts as an escorting protein to bring the Rab proteins. These are targeting proteins that are part of the membranes of cells. These need to undergo lipid modification so that they'll bind into the membrane and act as these targets. So if, with the eye of faith, if you look at lane 2, lane 2 is the control fibroblasts. This is a normal individual. lane 3, you'll see the very bright red in the lower part below the green band. You'll see that there's a significant amount of unprenylated or unlipid modified Rab proteins. And if you swing all the way over to lane 5, if you're reading from right to left, you will see that once you add the RNA, that's this AO1C, to the cells, it creates a profile that is similar to a normal individual in terms of the biochemistry. So from our standpoint, at least in our lab, we've tested multiple antisense oligonucleotides with 2 different chemistries. So this is modifying the RNA, and they have really increased the amount of normal protein that's present. They've provided biochemical evidence of a restored protein. And so the studies we need to do now are to take this from a cell level to validate safety in some model and manufacture this as a good manufacturing in a very specific way to manufacture this for delivery. And so I want to thank you for listening. And if you have any comments, you can get in touch with me in Montreal or at the University of Alberta addresses. And I hadn't put up a slide here with my funding because unfortunately I haven't had any funding to do this work. But that's my problem, not your problem. And so I'll sit down now.